मुझे बता देना। Hi everyone, this is Dr. Mamta Jawa, your microbiology faculty at PW MedEd, and in this session we are going to learn about a very important topic of general microbiology, and that is bacterial spores. Now guys, I personally believe that we can learn something or the other, some life lesson. Definitely we're going to learn from we can learn one or another important life lesson from any bacterium, virus, fungus, or even in immunology, and even from structures like spores. Now, the very first lesson I would want my students to learn from bacterial spores is the lesson of resilience. Now, first and foremost thing which you should remember about bacterial spores is that these are structures which are said to be highly resistant to adverse conditions or adversities which a bacterium might happen to face in its lifetime. So it is extremely resistant, or I would want to call it resilient. In textbooks, you would encounter this word resistant. Now, what exactly this structure resistant to? Now, the exact definition of bacterial spore I would highlight in just a few minutes. Before that, remember these are highly resistant structures. Resistant to what? Resistant to a lot of antibiotics, resistant to UV rays, and a variety of disinfectants. Okay, a variety of disinfectants. Now, as I said, these are resistant structures. They are highly resilient. They are able to, overcome adversities. Now, what happens? Not only human beings face challenges in life, even bacteria face a lot of challenges. Now, what challenges are we talking about? A bacterium could face a challenging situation in the sense that the nutrients might be low, or if it is an aerobic organism, an aerobic bacterium, it could encounter an area or domain where oxygen is not enough, or if it is an anaerobe, it could be exposed to traces of oxygen which could be detrimental to its life. So all of these situations, all of these conditions are said to be adverse conditions. So what happens? Under such adverse conditions, there is going to be a major life change, a major transformation of a vegetative bacterium into what is known as a spore. And these adverse conditions, as I already pointed out, it could mean a depletion of nutrients, or for aerobic organisms, it could mean a decrease in the content of oxygen around, or for anaerobes, it could be exposure to traces of oxygen. All right. Now, let's try to understand what exactly is meant by a vegetative bacterium. Alright guys, so over here you can see a happy-go-lucky vegetative organism, a vegetative bacterium it is. Now everything is going on absolutely fine in the life of this specific bacterium. इसकी ग्रह दशा बहुत अच्छी चल रही है, कोई problem नहीं life में, as in there is adequate availability of nutrients. If it is an aerobe, it is getting sufficient amount of oxygen. If it is an anaerobe, it is away from oxygen, which otherwise would be toxic for it. And I would want all of you to notice the shape I have drawn and the color of this happy-go-lucky vegetative bacterium. The color I have drawn is blue, and remember, by far the most commonly used differential stain anywhere in any microbiology lab the world over is gram stain. Now on gram stain, we categorize microorganisms into two categories: gram positive fellows and gram negative fellows. Gram positive organisms or gram positive fellows take up the blue color, which is the color of positivity, whereas gram negative organisms are going to take up red color, which reminds us of, you know what, kuch negative, khoon kharaba, war, bloodshed. All right, so this is how I used to remember gram reaction when I was a student. So gram positive organisms or gram positive structures are going to take up the color of positivity, that is blue color on gram stain, and gram negative organisms or gram negative structures are going to take up the color of negativity, that is the color red. Okay. All right. So this is the definition of a vegetative bacterium, where it is happily multiplying. And if I ask you, how do the bacteria multiply? Let me see some answers in the chat box. How do bacteria multiply? Is it mitosis? Is it meiosis? Or is it something else? All right, guys. Now, bacteria multiply by what? Bacteria multiply by the process of binary fission. Remember, B for binary fission, B for bacteria. So a happy-go-lucky or vegetative bacterium is the one which is happily multiplying because it is getting adequate nutrients, and the other conditions with regard to oxygen, carbon dioxide, and other availability of the other growth factors is very well available. All right, now under adverse conditions it will transform into a spore. So what exactly are bacterial spores? Remember, bacterial spores are structures which are going to be formed under adverse conditions, and bacterial spores are pretty thick walled. They are extremely thick walled structures. So let's say, you know, we have got a bacterium. Now, the reason why I chose blue color to draw the happy-go-lucky vegetative bacterium is because, as I said, blue is the color of positivity. That is, the gram-positive organisms are going to appear blue on gram stain. So remember, a basic rule about bacterial spores is that bacterial spores are produced only by gram positive organisms and not at all by gram negative organisms. This is the basic rule. There are teeny tiny exceptions to this, but at undergraduate level and from the point of view of your exams, be it NEET PG, INICET or FMG, just remember this basic rule that spores are produced by gram positive organisms, and that too not all gram positive organisms. And the important gram positive fellows which produce spores belong to the genera Bacillus and Clostridium. Okay, so B stands for genus Bacillus and C stands for genus Clostridium. Now both of these are what? So as I said, as a basic rule, we need to remember that bacterial spores are formed or produced only by gram positive organisms. So both Bacillus and Clostridium are what? They are gram positive bacteria, and these are what exactly shaped bacteria? These are gram positive rods, also known as gram positive bacilli. Okay, gram positive rods or bacilli. Now, one more thing I would want you all to remember in the context of BC is, remember, BC means what? BC should remind us of what? The fancy full form these days of BC is before COVID, but I don't want you all to remember before COVID. I would want all of you to remember that spores survive. Spores survive since BC. I would want you all to remember BC, that is before Christ, meaning to say that spores which I said are highly thick walled structures which are resistant to a whole lot of things, they are going to survive since BC, before Christ, meaning they are going to survive in nature for decades and decades and decades altogether. So they survive in nature for many years altogether. So remember, spore survives since BC, and also BC also serves as a reminder for the two important bacterial genera who are spore producers, that is genus Bacillus and genus Clostridium. Okay, genus Clostridium. So we saw how a happy-go-lucky or vegetative bacterium looks like. Okay. Okay. Education dark screen mode is right now not available. I hope the handwriting is okay for all of you. I hope it is legible. If you are unable to understand anything, please do let me know. Okay, so spore survives since BC. B for Bacillus, C for Clostridium. अब इसको देखो. All right. So what is going to happen under adverse conditions is that the happy-go-lucky vegetative bacterium is going to transform itself into spore. So I can give you a similar analogy of this process. The simple analogy which I can give you is the sleep mode becomes on. Just like, when we board a flight, we have to, keep our phones in the sleep mode so that, we are disconnected from the world, from the reality, I mean, especially the internet. And so that is kind of a break for us from social media. Okay. So similarly, this when a bacterium transforms into a thick walled structure, that is spore, the bacterium is going to shut down its metabolic activity. Now see this. Okay, guys. So a vegetative bacterium, when it changes into a spore, that is the sleep mode is on, this spore is going to have very decreased or less metabolic activity. Remember, the sleep mode is on. Now, why the metabolic activity is going to be less? It is going to have less metabolic activity because spores per se are extremely dry structures. They have got very less content of dihydrogen monoxide, in other words, water. Now guys, jal hi jeevan hai. All the biochemical reactions take place in the milieu or environment of water. So obviously, when I'm saying that spore is an extremely dry structure, it has got very less water content. Obviously, the metabolic activity is going to be low in spores. When the metabolic activity is going to be low, it is not going to be able to synthesize so many things, like no protein synthesis is going to be there, and exotoxins are a fantastic example of a very important kind of proteins produced by so many bacteria. So all the metabolic activity is going to be suspended, and because of this reason that the metabolic activity is going to be curtailed or toned down so much, there is going to be increased resistance to what? There is going to be increased resistance to antibiotics, because antibiotics are at their functional best when? Antibiotics are at their functional best when the metabolic activity of bacteria is maximum. ठीक है? All right, let me just change the color so that even if it is too bright, okay, let me not use this color. Just let me know if this color is okay. Guys, is this color okay? Maybe because I was using some that this color is fluorescent color. I hope this color is okay. Okay, so henceforth I'm gonna use this color. Do let me know in the comments. Serene. ठीक है, तो स्पोर को मैंने क्या बताया? इट्स एन एक्सट्रीमली ड्राई स्ट्रक्चर, तो मेटाबॉलिक एक्टिविटी कम होगी. मेटाबॉलिक एक्टिविटी कम होगी तो क्या होगा? रेजिस्टेंस टू एंटीबायोटिक्स ज्यादा रहेगी. एंड मैंने पहले ही बताया, दीज आर एक्सट्रीमली थिक वॉल्ड. दे आर एक्सट्रीमली thick walled structures, and one vegetative bacterium is going to give rise to or transform into one spore. This process is known as what? This process is known as sporulation. Okay, sporulation. So this by virtue of all these because of all these reasons, that it is extremely thick walled, and therefore it is going to also exhibit increased resistance to what? It is going to exhibit increased resistance also to a lot of disinfectants, okay, a lot of disinfectants and UV rays. So that is how the spore is going to be able to survive under adverse conditions. The process of transformation of one vegetative organism or bacterium into a spore is known as sporulation. So one vegetative organism or bacterium is giving rise to one spore only. Okay, so please remember, bacterial spores are not at all a method of multiplication of bacteria, guys. Bacteria कैसे multiply करते हैं, मैंने अभी थोड़ी देर पहले ही पूछा था. Bacteria multiply by binary fission. ठीक है, bacteria multiply by binary fission. All right. Now, okay, education, no metabolic activity, then how will it survive? Some basal enzymes which are needed to survive, which are, utmost essential for survival. Some nutrient reserves are there. So, some of these, enzymes, as I said, which are necessary for survival, they are still going to be active. They're still going to be active. ठीक है, तो मैंने लिखा है decreased metabolic activity, not zero activity altogether. Education, I hope that answers your doubt. ठीक है, so remember, bacterial spores unlike fungal spores are not a method of multiplication of bacteria. Now fungal spores on the other hand, fungal spores क्या है? Fungal spores are a method of multiplication of the fungi. Okay, multiplication of the fungi. Is this clear? ठीक है, तो चलो हमने देख लिया कि एडवर्स कंडीशंस पे क्या हो रहा है. कुछ बैक्टीरिया स्पोर बना रहे हैं. विच बैक्टीरिया आर दोज? दे आर ओनली ग्राम पॉजिटिव ऑर्गेनिजम्स. शेप वाइज दे आर रॉड्स और बैसिलाई. विच आर दोज बैक्टीरिया? रिमेंबर, स्पोर सर्वाइव सिंस बीसी. तो बीसी से आप याद रखोगे, बी फॉर बैसिलस जीनस, सी फॉर क्लोस्ट्रीडियम. ठीक है, तो अब क्या होगा, देखो रात के बाद दिन आता है. हमेशा टाइम खराब नहीं होता ना लाइफ में. रात के बाद जैसे मैंने बोला दिन आता है. एडवर्स कंडीशंस बैक्टीरिया की लाइफ में भी हमेशा नहीं रहेंगी. इसकी भी ग्रह दशा कभी तो टुवर्ड्स पॉजिटिविटी टर्न लेगी. सो व्हाट आई एम ट्राइंग टू से इज, ऑन द रिटर्न ऑफ फेवरेबल कंडीशंस, व्हाट इज गोइंग टू हैपन इज, ऑन द रिटर्न ऑफ favorable conditions. Do let me know if this font is working in this much brightness. I hope, at least according to me, it is more legible than before. You please tell me if you are okay with this. On return of favorable conditions क्या होगा? स्पोर्स जो हमने इतना थिक वॉल स्ट्रक्चर बनाया था ना, ये देखो स्लीप मोड ऑन था, ये सो रहा था. अब ये वापस से क्या बन जाएगा? अब ये वापस से हैप्पी गो लकी वेजिटेटिव बैक्टीरियम बन जाएगा. और इस प्रोसेस को हम क्या बोल देंगे? इस प्रोसेस को हम बोल देंगे जर्मिनेशन ऑफ स्पोर्स. तो जर्मिनेशन का क्या मतलब हुआ? जर्मिनेट वर्ड का क्या मतलब हुआ? जर्मिनेशन मीन्स टू गिव राइज टू समथिंग, किसी एक्टिव चीज को जन्म देना. ठीक है, सो क्या होगा? रिटर्न ऑफ फेवरेबल कंडीशंस. जैसे कि अगर पहले न्यूट्रिएंट्स डिप्लीटेड थे तो अब वो मिल जाएंगे. अगर एक एरोबिक ऑर्गेनिज्म था जिसको एनफ ऑक्सीजन नहीं मिल रही थी, अब उसको वो भी मिलने लगेगी. सो बेसिकली जो चीजें पहले खराब चल रही थी अब वो ठीक हो जाएंगी. सो ऑन द रिटर्न ऑफ फेवरेबल कंडीशंस or going away of adverse conditions, the sleep mode on wala structure, that is bacterial spore, is going to be transformed into the happy go lucky vegetative bacterium. और इस process को आप क्या बोलोगे? Germination. So these are very very important terms guys. Germination क्या होता है? Sporulation क्या होती है? Is this clear? Is this clear? Let me know in the chat box. चलो नेक्स्ट टाइम वील ट्राई कि इसी सॉफ्टवेयर में हम डार्क मोड भी यूज करें. All right. Now, next thing about bacterial spores which I would want all of you to remember is किसने discover की थी? Now, remember, bacterial spores पहली बात तो किसने डिस्कवर की थी एंड किस में डिस्कवर की थी? किस बैक्टीरियम में? बैक्टीरियल स्पोर्स वर डिस्कवर्ड इन कौन सा बैक्टीरियम? Can someone in the chat box let me know which is the biggest bacterium in the entire clinical bacteriology? Let me know. सबसे बड़ा बैक्टीरियम कौन सा है भाई? ये आपके लिए एमसीक्यू भी है और इससे एक दो चीजें रिलेटेड मैं और भी बताने वाली हूं. My question is guys, एक्सेलेंट, आर जगदीश्वर, बैसिलस एंथ्रासिस, वंडरफुल। सो सबसे बड़े बैक्टीरियम का नाम क्या है भाई? चलो ब्लू में लिखते हैं उसका नाम। कोई बता सकता है ब्लू में ही क्यों लिखा मैंने? नॉट बिकॉज ब्लू मेरा फेवरेट कलर है, वो तो है ही, बट उस रीजन से आई एम नॉट राइटिंग इट इन ब्लू। द रीजन इज बैसिलस एंथ्रासिस, मैंने अभी बताया था ना, बैसिलस जीनस क्लोस्ट्रीडियम, ये क्या है? ग्राम पॉजिटिव रॉड्स हैं। सो दिस इज अगेन अ ग्राम पॉजिटिव रॉड, तभी मैंने नाम ब्लू में लिख दिया। सो इट्स अ ग्राम पॉजिटिव रॉड और अ ग्राम पॉजिटिव बैसिलस। दिस इज द बिगेस्ट बैक्टीरियम। सो गाइस इमेजिन करो कि इतना बड़ा बैक्टीरियम था, यही सबसे इट्स वन ऑफ द मोस्ट मेडिकली सिग्निफिकेंट बैक्टीरियम। दिस वाज द बैक्टीरियम व्हिच वाज फर्स्ट सीन अंडर द माइक्रोस्कोप। तो इमेजिन ये इतना बड़ा था कि ये सबसे पहले दिख गया माइक्रोस्कोप में। इसका साइज कितना होता है? 4 to 8 बाय 1 to 1.5 माइक्रॉन्स। ठीक है, सो इसको ऐसे याद करो, ये इतना बड़ा था, सबसे बड़ा था कि इट वाज फर्स्ट एवर बैक्टीरियम व्हिच वाज सीन अंडर द माइक्रोस्कोप वाज दिस बैक्टीरियम। और किसने इसको देखा था माइक्रोस्कोप के अंदर? पोलेंडर। सो रिमेंबर, पोलेंडर, एंड में क्या आ रहा है? ENDR, पोलेंडर सॉ इट अंडर द माइक्रोस्कोप। और यही इतना बड़ा जब बैक्टीरियम हम इसको बोल रहे हैं, तो यही वो बैक्टीरियम है जिसके कंटेक्स्ट में, जिसके केस में स्पोर्स भी डिस्कवर हुई। तो ये इतना बड़ा था कि इसकी स्पोर्स भी सबसे पहले देखी गई। सो बैक्टीरियल स्पोर्स वर डिस्कवर्ड इन बैसिलस एंथ्रासिस। और किसने डिस्कवर की थी? डिस्कवर्ड बाय, द स्पोर्स वर डिस्कवर्ड बाय सर रॉबर्ट कॉक, who is appropriately regarded as whom? He is appropriately regarded as the father of bacteriology, because he is endowed with the he is credited with the discovery of so many important bacteria. ठीक है, so spores were discovered by Sir Robert Koch in Bacillus anthracis, which is the biggest bacterium. साइज कितना है इसका? 4 to 8 बाई 1.5 1 to 1.5 माइक्रॉन्स, ये रेंज हो गई। एंड रिमेंबर गाइस, जब हम कह रहे हैं ना कि इतना बड़ा बैक्टीरियम है, तो इसकी ईगो भी बहुत बड़ी है, और इसको मैंने डिटेल में PW मेडेड के लेक्चर्स में डिस्कस किया है कि व्हाई एम आई सेइंग इट इज एन ईगोइस्टिक ऑर्गेनिज्म। देयर इज अ वेरी easy way to remember the salient differences between this organism Bacillus anthracis and the other species of genus Bacillus which are known as anthracoid bacilli, जो आप PW मेडेड के लेक्चर्स में देख सकते हो। अब बहुत ईजी है, बुक्स में यूजुअली टेबल दिया होता है 10-11 पॉइंट्स का। आई हैव ट्राई टू सिंप्लीफाई इट बाय calling this organism as an egoistic fellow, तो बहुत easy आप लोगों के लिए याद करना। So do check out that video. It's not a lengthy video. It's very very important, especially for second year people. You can be asked questions on the differences in your viva exams, और theory paper में तो आता ही आता है। ठीक है, सो बैसिलस एंथ्रासिस में हमने देख लिया कि स्पोर्स डिस्कवर हुई थी। एंड रिमेंबर आई वाज सेइंग कि ये सबसे बड़ा बैक्टीरियम है, तो इतना बड़ा है, इसकी ईगो इतनी बड़ी है कि ये नॉन मोटाइल है, अपनी जगह से नहीं हिलता। सो रिमेंबर, द बिगेस्ट बैक्टीरियम इन द एंटायर क्लिनिकल बैक्टीरियोलॉजी क्या है? ये नॉन मोटाइल है। इस वाला मैंने ऑफ द कॉन्टेक्स्ट फ्रॉम बैक्टीरियल स्पोर्स से जाके आपको बता दिया। ठीक है, सो रिमेंबर दिस। सो बैक्टीरियल स्पोर्स अभी तक हमने क्या देख लिया कि कब बनती हैं, बनने को क्या बोलते हो? स्पोर्यूलेशन। वापस से जब स्पोर से बैक्टीरियम बन जाता है तो उसको जर्मिनेशन बोलते हो। एंड थिक वॉल्ड होती हैं, ड्राई होती हैं, ये तो ये भी रटने की बात नहीं है। अगर तो ड्राई है तो मेटाबॉलिक एक्टिविटी कम होगी, जब मेटाबॉलिक एक्टिविटी कम है तो क्या होगा? इंक्रीज रेजिस्टेंस टू एंटीबायोटिक्स होगी। एंड बिकॉज ऑफ इट्स वेरी स्ट्रक्चर, बहुत थिक वॉल्ड स्ट्रक्चर होता है, इट इज ऑल्सो रेजिस्टेंट टू होल लॉट ऑफ डिसइंफेक्टेंट्स एंड यूवी रेज ऑल्सो। एंड बट रिमेंबर, दीज आर नॉट द मोस्ट रेजिस्टेंट स्ट्रक्चर्स, विच इज बाय फार द मोस्ट the structure which is notoriously resistant to a whole lot of things, a whole lot of disinfectants, UV rays बोल दो, एंटीबायोटिक्स तो खैर वैसे ही काम नहीं करती हैं, which is overall in the hierarchy of susceptibility to different disinfectants and antiseptics etc. कौन सा स्ट्रक्चर ओवरऑल सबसे ज्यादा रेजिस्टेंट माना जाता है? कैन समवन प्लीज टेल मी इन द कमेंट सेक्शन। प्रायोरिटी हम उसको देते हैं, नंबर 1 बोलते हैं। देखो मैंने तुम्हें अभी हिंट भी दे दिया, आई हैव टोल्ड यू कि उसको हम प्रायोरिटी पे रखते हैं, मतलब नंबर 1 पे रखते हैं अमंगस्ट ऑल द थिंग्स व्हेन वी ट्राई टू ग्रेड देम इनटू कौन सा सबसे ज़्यादा ससेप्टेबल है टू डिसइंफेक्टेंट्स, फिर कौन सा उससे ज़्यादा रेजिस्टेंट है, फिर मोस्ट रेजिस्टेंट कौन सा है। प्रायोरिटी एजेंट, लेट मी सी हाउ मेनी ऑफ यू आर एबल टू गेस द नेम। from this word priority? कुछ याद आ रहा है इसको देख के? अब तो मैंने ऑलमोस्ट तुम्हें नाम बता ही दिया है, इससे कुछ भी याद नहीं आ रहा? PRIORIT, भाई PRI से कुछ सोचो. Some important, I won't say organisms, but something, some important topic you read in microbiology, clinical microbiology, because rare human diseases. Anyone? या मैं बता ही दूं? Guys, I'm talking about prions. ठीक है, prions. Prions are क्या क्या होते हैं? प्रोटीन्स होते हैं, प्रोटीनेशियस इन्फेक्शियस पार्टिकल्स. PRI, वेरी गुड, अब तो मैंने बता दिया. प्रायन सबसे ज्यादा रेजिस्टेंट होते हैं, तो सेकंड मोस्ट रेजिस्टेंट स्ट्रक्चर्स होते हैं हमारे बैक्टीरियल स्पोर्स. ठीक है, तो इस चीज को याद रखना. एंड देन वन more important thing about bacterial spores is that, you know, bacterial spores have got what is known as dipicolinic acid. ठीक है, तो spores में क्या होता है? Spores have got this chemical, namely dipicolinic acid. And what is this and how is it going to help bacterial spores survive? Dipicolinic acid is a chelator of calcium ions. These are what? These are divalent cations. ठीक है, तो अगर एक वेजिटेटिव ऑर्गेनिज्म या वेजिटेटिव बैक्टीरियम इज एक्सपोज टू दीज डाइवेलेंट केटाइंस लाइक कैल्शियम आयंस, the bacterium is going to be harmed. So remember, as the rule, these divalent cations like calcium ions mostly are going to harm a vegetative bacterium, but in a spore these are going to be rendered useless because the spores have got what is known as dipicolinic acid, which is going to chelate calcium ions. In other words, chelation का क्या मतलब है? To form a complex. टू फॉर्म अ कॉम्प्लेक्स. तो ये शील्ड या ढाल की तरह एक्ट करेगा. अगर तो कैल्शियम आयंस प्रेजेंट है इन द विसिनिटी ऑफ बैक्टीरियल स्पोर, तो क्या होगा? These are going to form a complex with the calcium ions, and that is why calcium ions will become busy with dipicolinic acid and they won't be able to harm the spore. Is this clear? Is this clear everyone? सो ये भी आपको याद रखना है, डिपिकोलिनिक एसिड प्रेजेंट होता है बैक्टीरियल स्पोर्स में. If you have any doubts so far, do let me know. Now, a very good analogy which I can give you to understand the basic concept behind bacterial spores is the seed of a plant. Those of you who love gardening, you would obviously know कि भाई seed क्या है. कोई plant का जो seed होता है, seed of a plant. Seed of a plant is very very analogous or similar to bacterial spores. बैक्टीरियल स्पोर्स, क्योंकि जस्ट लाइक बैक्टीरियल स्पोर्स आर एक्सट्रीमली ड्राई स्ट्रक्चर्स, सीड ऑफ अ प्लांट भी क्या है? ये भी बहुत बहुत ड्राई होता है. इट्स अ वेरी वेरी ड्राई स्ट्रक्चर. लेकिन जब आप इसको एक्सपोज करते हो टू यू नो गुड कंडीशंस, इसको आप जब सॉइल में प्लांट करोगे, इसको पानी प्रोवाइड करोगे, इसको दैट इज इट इज व्हेन इट इज वाटर्ड, इट इज गोइंग टू बी ट्रांसफॉर्म्ड इनटू व्हाट? इट इज गोइंग टू बी ट्रांसफॉर्म्ड इनटू अ ब्यूटीफुल प्लांट. अ ब्यूटीफुल प्लांट. सो उसी तरह से a bacterial spore also on germination is going to give rise to what? It is going to give rise to a vegetative bacterium. ठीक है, it is going to give rise to a nice happy go lucky vegetative bacterium. So this is a very good analogy which you need to remember. ठीक है. Now spores की भी अपनी shapes होती हैं और अपनी अपनी positions होती हैं central position, subterminal and all of that I have discussed in detail under the video modules on Bacillus and Clostridium. You can find these on under the video lectures on PW MedEd. And another thing, and probably the last thing for today's session regarding bacterial spores, I would want all of you to remember is that, see, bacterial spores are not stained. They are not stained on gram stain. Gram stain जिसको हम बोलते हैं, it is the most commonly used differential stain all over the world, whether it's a PHC lab or it's a tertiary care hospital. This stain also is unable to stain the bacterial spores. ठीक है, तो bacterial spores अगर आप तो क्या होगा, gram stain पे bacterial spores are going to be unstained. So bacterial spores are going to be seen as unstained areas on gram stain. अगर आप चाहते हो spores को stain करना तो आप क्या कर सकते हो? Remember bacterial spores are what? They are acid fast. They are acid fast structures और इनकी staining के लिए you can therefore use 0.25% sulfuric acid during as the decolorizer during Zn staining. And अगर आप 25% ऑफ H2SO4 के अतिरिक्त उसके अलावा कोई भी कॉन्सेंट्रेशन ऑफ H2SO4 दैट इज डीकलराइजर की यूज करते हो, उसे क्या बोलते हो आप? यू कॉल दैट स्टेनिंग प्रोसीजर एज मोडिफाइड Zn स्टेनिंग. ओके, एज मोडिफाइड Zn स्टेन. ठीक है, एंड इसके अलावा बैक्टीरियल स्पोर्स को स्टेन करने का एक और अच्छा तरीका क्या है? एक और अच्छा तरीका है स्पोर स्टेन पर से, नेमली मैलाकाइट ग्रीन. ठीक है, malachite green is referred to as or deemed as the spore stain. ठीक है, so do remember this. All right, any doubts you have so far, please feel free to ask. Any doubts, anyone? Any doubts? सो बैक्टीरियल स्पोर से क्या टेक होम मैसेज लेना हम चाहेंगे? हम ये मैसेज, आई मीन दिस इज व्हाट आई वुड वांट to learn myself from bacterial spores, and I would want my students also to learn that कभी भी adverse conditions life में आती हैं तो you have to be tough, you have to be resistant, you have to be resilient. ठीक है, you should not give up because the conditions are not always going to be adverse. So just keep at it, hang in there, and all will be well. So on this note, I would want to end this session over here, and I hope to see you all in the next live session, which hopefully is going to be conducted next week. And the topic, I suppose, is actinomyces. And in case you have any doubts, please let me know in the comment section. Please check out the description of the video of this, the description of this particular video to find out all the social media handles of PW MedEd. We have got a Telegram group and also a Facebook group where all of us are quite active. You can post your queries, you can post your doubts and tag the faculties over there. If you want to personally connect with me, you can follow me on my Instagram handle, that is mj.micro. I'm most active on Instagram, not I'm probably on Telegram also, but on the groups, not DMs. ठीक है, if you want any kind of help from me, you can connect with me, you can DM me on mj.micro. So guys, on this note, I would want to end this session, and I thank all of you for your patient listening. Do let me know your comments in the comments section about this session, and if you want us to conduct more such YouTube sessions, not just this but anything you would want to do on YouTube, please do let us know. And let me also tell you, we are going to be coming up with more integrated sessions also in a very short span of time. So keep studying. God bless you all for not just exams but everything else in life. Thank you.